of ours. Right, well, so the recording was started. So I was just saying welcome to our audience for joining us today with another installment of our CCPBioSim series of industry presentations. We try to have one per month. And today we are delighted to welcome Márton Vass as our speaker from Schrödinger. So Márton is currently a principal application scientist at Schrödinger Technologies. He studied chemistry and chemoinformatics in Budapest, Hungary, and was fortunate to start working at Gedeon Richter, where in fact he did his PhD in computational fragment-based drug discovery. Gedeon Richter was then a mid-sized pharmaceutical company also in Budapest. And after his PhD, Márton did a postdoctoral training at Vrije University in Amsterdam, also working on chemoinformatics methods for GPCR drug discovery. And after that, he moved to London and worked at Benevolent AI on machine learning for compound design. So a very broad range of experience. And currently, so Márton has been with Schrödinger for almost 3 years, and his research is focused on large-scale free energy perturbation calculation in live design and testing the usability of predicted protein structures in drug discovery. Today, his title is on, you can see the title of the talk on the slide, so I'm not going to say that. Welcome, Márton, and thanks again for making time for this presentation.
Thank you very much for the introduction, and especially thank you for the invitation to give this talk. So yes, actually you've pretty much covered this slide that I created. So maybe I'll just say a few words about the roles I had in each of these places where I worked and did my PhD and postdoc studies. So at Gedeon Richter is an industrial pharmaceutical company. And of course, as a starting modeler, I had to support several drug discovery projects. But I was lucky enough to do my PhD parallel to my regular work. And my boss at the time was also my PhD supervisor. So I'm really grateful to him. I worked quite a lot on membrane protein targets, so GPCRs and ion channels, and that kind of continued throughout my my career. And that's how I got into Amsterdam, into the group in Amsterdam, where the focus was obviously on aminergic GPCR modeling as well. But what I learned in Amsterdam was really cross-functional teamwork. We had a little team of 5 people who were medicinal chemists, modelers, software developers, and someone who was also an expert in KNIME, which is a kind of like a graphical programming interface. So this is where I learned a lot about working together with different disciplines and especially software developers on developing new methodologies. And later on in Benevolent AI, I picked up a lot about machine learning, of course. Being, of course, also a pharmaceutical company, I, of course, had to support some drug discovery projects. But also I was leading a small group of 7-8 people, and we were working on machine learning models where we took not only the ligand structure, but the protein context also into account. So that was a very exciting thing to do. And that's where my interest with AlphaFold and machine learning protein structure prediction comes from mainly. And then in Schrödinger, since I joined, of course, I work with our customers, and that's the main focus of our work. But my interest in AlphaFold and other machine learning protein structure prediction methods has continued, and I try to be involved in all of this research that is carried out within Schrödinger. And related to that, of course, FEP is one of our flagship products. So that's free energy perturbation for anyone who doesn't know that. And this makes quite a good kind of bundle of research because they are very, very tightly related to each other, as I hope I will be able to demonstrate in my talk. Schrödinger is a global company. We are present either with offices or just remote teams in a lot of different countries and continents. And we have various software suites supporting small molecule drug discovery, materials science, which is actually a very exciting field. And if anyone doesn't know what Schrödinger is working on in this field, I recommend checking out our website because we have some very exciting applications there. We also support other modalities, biologics, and nowadays RNA drug discovery and proteins and other types of targets. And live design, of course, is our discovery enterprise informatics tool, and PyMo, which is our high quality visualization software. If you are using PyMo, I suggest checking out PyMo Free, which has just recently come out to generate stunning videos and images of your molecules. So as an application scientist, what I mainly do is provide scientific support to our current and prospective customers, demonstrating the optimal use of our software, but also taking the feedback of our customers and feeding it back to our product development, and vice versa. If anything comes out fresh or hot off the press, we introduce those things to the customers and try to get feedback. And in general, we try to be respectable scientists and give not just guidance about our software, but general scientific guidance to our customers as well. And in order to do that, we do hopefully cutting-edge research in Schrödinger, both method development in the company and working together with our customers. There is an interesting blog post about the work of an application scientist and how that might not be exactly what people think an application scientist is. So I really recommend reading that if you have the time. But today I'm going to talk about the science that we are working on, and especially the topics that I have been part of inside Schrödinger, and that is AlphaFold, using AlphaFold structures for virtual screening, for enabling targets, specifically GPCRs in this case, and common ADME targets for structure-based drug discovery or structure-based modeling. And in the meantime, while I go through my slides, I will introduce some of the models and methods that we employ to do this at Schrödinger. So how does AlphaFold come into the picture of drug discovery? When you have a new target, you can either choose to go down the ligand-based route if you don't have any clue about how the protein looks like or how your target looks like. In that case, you can use machine learning, ligand-based methodologies such as shape screening, pharmacophore screening, and other tools. But let's say you want to do structure-based modeling, which is becoming more and more available for targets that are crystallized in the PDB, or have been an experimental structure have been deposited in the PDB, or you can create a homology model or download an AlphaFold to model, which are not currently quite easily available. But an AlphaFold model is not always a great starting point or sufficiently good starting point for the modeling work that we want to do. So you might need to do some extra work to refine the structure and to be able to generate a binding mode that is then amenable to further modeling, either with free energy methods or other methods that we use for lead optimization. A very quick overview, but I'm sure everyone has heard a lot about AlphaFold and related methodologies, especially in the last few years. They have got quite a lot of publicity, but maybe it's quite interesting to know that the residue coevolution concept was introduced around 1994, so quite a bit of time ago now. And even some structure prediction methods have been devised in the early 2010 decade. But then, of course, a big improvement or big leap started happening in CASP in the late 2018, when AlphaFold from DeepMind won the CASP 13 competition and demonstrated that this concept and method is actually useful for protein structure prediction and more useful than previously other tools were. But of course, in the great breakthrough happened in 2020 when AlphaFold 2 came into the picture and won the CASP 14 competition, by far above everyone else. And then immediately they published the database with 21 genomes and proteins predicted for 21 genomes. And also they published the code, which then created a boom of other methods that were developed based off of AlphaFold. And then a lot of people started working on this and started developing other methods similar to AlphaFold or based on AlphaFold. And AlphaFold Multimer came. The database was extended to about 200 million protein structures. And AlphaFold 2 based models won again in the 2022 competition of CASP 15. Another maybe interesting thing is that Meta came out with the large language model based protein structure prediction and a database of about 600 million proteins as well. And on the bottom of the timeline, I just want to mention that machine learning docking methods have been evolving alongside these models. And more recently, AlphaFold latest, RosettaFold, OLATEM, and some other methods have been attempting to co-fold proteins and ligands, which I'm quite curious to see where this effort goes. But most of these methods are not yet open source or not yet very well tested. So of course, we want to look at the targets where we don't have a crystal structure, but we do have ligand data. And if we plot the number of compounds in Campbell, which is a biochemistry database, against the number of PDB structures, we can see that there are quite a few structures without with this kind of missing structural information. And if we take a look at some of those top targets, we can see that quite a few of them are GPCRs and membrane proteins. So right up my alley. So that's why at Schrödinger, we of course were interested in looking at these structures, and our first effort was to look at them in terms of virtual screening. So in order to collect our dataset for a benchmark, we looked at the DADI dataset, which is a very well established dataset of active and decoy ligands. Maybe not the best one. There are others out there, but maybe the oldest and most well documented and maintained one. So we looked at 28 targets that were amenable to our workflow. Many of them kinases, but also proteases, nuclear receptors, and some other targets in that as well. Unfortunately, there were some targets which were not amenable, either because they contained covalent ligands or severe clashes of the AlphaFold structure with the ligand binding site, which can be manually kind of resolved, but we really wanted to look at an automated workflow in this case. We also had some targets with cofactors, and we treated those somewhat separately. But I'm going to talk about mainly those 28 targets that were amenable for MD simulations. So one thing to remember about AlphaFold structures is that they predict the structure for the whole sequence of the protein, whether that is whether the whole sequence is ordered or intrinsically unordered. Sorry, 1 second. Sorry, I'm back. So AlphaFold will give you a prediction for every single residue in the protein, but some of those have a very low, very high uncertainty, and sometimes these may overlap with the binding site. So we needed to trim the AlphaFold structures to the part of the protein which is actually kind of the useful part, or a single domain where the binding site is located. So we also developed some automated methods for trimming AlphaFold structures based on the predicted uncertainty of the residues. The virtual screening itself was according to best practices at Schrödinger. So starting with protein preparation, using sitemap for characterizing the binding pocket, IFD-MD, which is induced fit docking with MD simulation, was used to optimize binding sites. I'm going to talk much more about this later. Lig prep was used to prepare our ligands at pH 7. Glide was used to dock the ligands into the receptor structures, either into a single receptor or an ensemble of protein structures from the IFD-MD refinement. And finally, we calculated the usual properties or results from virtual screening, such as enrichment factor at 1% of the ligand set and area under the receiver operating characteristic curve. So here are some numbers, but what they mean is that basically among these 28 targets that we investigated, on average, the AlphaFold structures that we download from the database perform quite similarly to an APO structure. So an APO structure is one where the protein is crystallized as it is without any ligands. And sometimes these binding pockets are not very well formed for ligand binding. And it was maybe not so surprising that an AlphaFold structure as it comes out of the database performs similarly to these APO structures where the binding site is not really optimized for ligand recognition. We can also see that if we don't truncate the AlphaFold structures, so that's the AlphaFold to full row, sometimes we cannot even dock all of the ligands in our dataset. But if we truncate the structures, then the percentage of the docked ligands goes up again. So now IFD-MD is our secret sauce at Schrödinger, at least for this application. It has 2 tracks of calculations. On the CPU, we perform pharmacophore-guided docking and optimization of the binding pocket residues around the ligand binding mode. This is an iterative process, repeated several times. And then on the GPU, we calculate a water map calculation of the protein, which looks at water positions in the binding pocket and their thermodynamic parameters. After this, we use our W score scoring function, which takes into account whether a ligand can replace high energy water molecules or form maybe interactions with those waters in the binding pocket. We also perform metadynamics, ligand unbinding metadynamics from the protein to see how stable the protein ligand interactions are in the binding site. And we take all of this together, and finally we come up with a composite score called IFD-MD score that is made up of the glide score, the W score, and the metadynamics score together. So relative to our traditional IFD technology, which only used the CPU track of the previous method that I explained, the addition of the molecular dynamics steps increases the accuracy of sorry. So it increases the accuracy of the methodology greatly. We have demonstrated that this method works on a variety of protein target classes as well, and most of the times we can find the correct binding mode, the crystallographic binding mode in the top 2 poses predicted by IFD-MD, even for chemical series that are dissimilar to the to previously known co-crystal ligands. So once we optimize AlphaFold structures with this IFD-MD methodology, we can optimize the binding sites to be more similar to a binding pocket that is preformed for ligand recognition, and we can bring up the enrichment factors to a level very close to those of holo structures in the PDB. So holo structures are the ones which already have a ligand co-crystallized, and therefore the binding site is already organized for ligand binding. So one example that sorry, just one. Okay. So One example is EGFR. So this is a simple example of a kinase, but of course we had different target classes as well. But maybe it's easy to demonstrate the effects of IFD-MD on this target. So if you look at the AlphaFold structure in blue on the bottom left, you can see that if we use the full structure, which has a loop that overlaps with the ligand binding site, we get almost no enrichment whatsoever. So enrichment factor should be usually well over 1, but in that case we get only 1.5. Whereas if we truncate the AlphaFold structure, we get an enrichment factor of 11, which means that we find 11 times the number of active molecules in our ligand dataset than with random selection. In an apo structure of this target, which is the yellow structure on the top left, we observe a similar enrichment factor around 14, where the binding site is not really organized to recognize the ligand. In a holo structure on the top right in green, we get an enrichment factor of almost 30, which means that we get quite a good recognition of the active molecules. And if we optimize the binding site of the AlphaFold structure with IFD-MD, so that's the red structure on the bottom right, we get an enrichment factor of 21, so much better than the original AlphaFold structure, close to the holo, but not completely, not exactly the same, but close to the holo structure. So after this, we were quite encouraged to go on with our investigation of AlphaFold structures. And we were wondering whether we can actually not only find hits, but also optimize compounds using an AlphaFold structure. So this is where my previous research on GPCRs came in handy, because we had to enable IFD-MD on membrane targets as well. And of course, GPCRs are one of the biggest target classes for that. And we did a big benchmark on 82 GPCR targets, which are GPCR structures, which belong to 10 different receptors, covering several GPCR target classes. In this case, success was defined as a ligand RMSD less than 2.5 angstrom to the crystal structure, and found within the first or the second top scoring IFD-MD pose. So you can see that with IFD-MD, in 95% of the cases, we find the correct pose within the top 2 ranked poses, whereas with Glide, which is our regular docking methodology, we only find about half of those cases. So this was quite a good starting point. But then, so this was obviously on crystal structures, and then we were wondering whether we can also do this for homology models or AlphaFold structures as well. So at the time, we had so this was performed maybe started about 2 years ago, when I joined, or not soon after I joined Schrödinger. We had an interest in a target because of one of our collaborators. So we looked at the somatostatin receptor target class, or target family, and we set out to model SSTR4, SSTR2, and SSTR5. And I'm going to talk about SSTR4 because this is the one that I actually personally worked on myself. So we used AlphaFold to generate 5 different conformations of the initial receptor structure. And of course, you can see that the termini have low confidence. So we truncated those from the structure. And we had a Boehringer-Ingelheim patent from 2016 that had 79 ligands with agonist efficacy data ranging from sub-nanomolar to 2 micromolar ligands. We used IFD-MD to dock one ligand from the patent, the most active ligand from the patent into the structure. And then we used a subset of 64 ligands aligned to that ligand structure or pose generated by IFD-MD and performed FEP, so free energy perturbation on the aligned ligands to validate the IFD-MD poses. And the hypothesis was that if we can generate a binding mode that is close to reality, we will observe a good FEP accuracy, whereas any other model that is not close to the real biological binding mode, we would not see good FEP accuracy, because we cannot, in those cases, we would not be able to recapitulate the SAR of the ligands. So our workflow here was to, as I mentioned, generate 5 AlphaFold models and run induced fit docking on each of those models, which created or generated 5 times 5, so 25 possible poses to start from. Now, this is quite a lot of different poses and protein ligand complexes to run FEP on. And that's why we initially started with a smaller set of ligands to save some calculation. So in this case, it was 18 ligands that we started from. And as you can see in the table in the middle, most of those 25 complexes did not produce a good FEP correlation at all. The only 2 AlphaFold structures that we saw some sigma from was the AlphaFold rank 4 and rank 5 structures. But rank 4 was a middle, so somewhat good but not great FEP result, whereas the 2 pose 1 and pose 4 in the AlphaFold rank 5 structure provided quite good correlation and low errors. We also ran absolute binding FEP on some of the most active ligands from this set. And they kind of confirmed that out of those 2 protein ligand complexes, the most probable one is the AlphaFold rank 5 protein model with the IFD-MD pose number 1. So we were quite happy with this result. We got a good FEP correlation on a smaller ligand set that also was validated with ABFEP. But we, of course, still had to validate it with the full set of 64 ligands, as I mentioned before. And this is what we did in the end. And we still got a good recapitulation of the SAR, where the R-squared was close to 0.5, which is, it might not sound very high, but for a GPCR, this is actually, or a membrane protein, this is actually quite good correlation. And you can see that most of the prediction are within the 1 kcal per mole error bar. So we were quite positive about this result, and also looking at the interactions, we can see that the basic amine of the ligand is interacting with an aspartate negatively charged residue in the protein. The amide group is interacting with glutamine in the protein, and there is also a water bridge to a backbone carbonyl in the extracellular loop 2. And this 6+6 aromatic ring system is contained in a very hydrophobic subpocket of the binding site. We could also recapitulate the SAR with so-called single edge FEP. So the difference to the previous slide is that in this case, we do the perturbations just one by one from a single structure mutating to a related side chain, and we don't have cycles in this graph, whereas in the previous slide, we would have all the compounds connected to all the compounds, and we can employ also cycle closure corrections in FEP. But the indication that we get even a good correlation without cycle closure corrections means that this binding mode is probably quite correct, and just changing this naphthyl group to different 5+6 and 6+6 ring systems is possible with FEP. That's a great indication that the model is correct. So what did we actually have to do to achieve this model? So the only significant motion of the residues that we need to consider in the AlphaFold structure is leucine 297, which has to move out of the way to make space for the ligand. But provided that we start from the right AlphaFold model and the right protein conformation or backbone conformation of the protein, that is the tricky part. So we really need both components in the workflow to generate an initial protein conformation set and the induced fit docking model to consider the movement of the residues to accommodate the ligand. Later on, there was a cryo-electron microscopy structure of SSTR4 published, which contains this J2156 ligand, which is not exactly congeneric, but maybe we can identify some common pharmacophore elements with our ligands, such as the bicyclic aromatic ring and the basic amine interacting with the acid. And running IFD-MD on both on this Cryo-EM structure provided us with a very, very similar structure and a very good FEP performance for those ligands. And therefore, we concluded this that the agreement of these poses is quite a good indication that they should probably be correct. Sorry. So one thing to note is that so far I've always mentioned that this amine is charged and interacting with the aspartate in the binding site. But this is not always a given thing and not always immediately evident when we look at the ligands. And indeed, if we calculate pKa's with an accurate quantum quantum chemistry method for this amine, we can actually see that some of them are quite far away from or quite low, which means that they exist in both charged and uncharged states in solution. However, in the binding site, it's probably only the charged state that can bind strongly because of the ionic interaction with the protein. So this discrepancy, of course, means that there should be a correction to the binding affinity if we only consider one of the states in our FEP calculations. So we tried, so at Schrödinger, the general or best practice for FEP is to always consider the different states. But we just wanted to see what happens if we don't do this correction. And we performed a similar calculation as I showed before, without the FEP, without the pKa corrections. And in this case, as you can see, AlphaFold model 5 still performs okay, but as you can notice, it's less good than it was before. And actually, the AlphaFold number 2 model performs somewhat better. In this case, in the FEP calculations, if we don't consider the pKa correction. However, this binding mode turned out to be quite different from the binding mode that was validated. So, so this shows how important it is to actually consider all different protomeric and tautomeric states of your ligand in these calculations. So one thing to note is that there are, of course, other methods to generate protein ligand complexes for new chemical series, and one of those is obviously what we are trying to do, to generate protein structures, conformations, and then try to place the ligands into the binding site. But nowadays, there are a few of these machine learning models that try to fold the ligand and the protein together at the same time. So there is AlphaFold latest from DeepMind. There is DragonFold from the company called Charm Therapeutics. These are closed source code for now. And there is also Rosetta Fold all atom by the Baker Lab, which is more accessible than the other two currently. So I'm really curious to see where these methods will go and how accurate they will be for new protein ligand complexes. At least in my hands, so far, some of these methods have not worked as well as I would have hoped, but we will see where we go with them. And finally, in the last few slides, I would like to talk about the extension of this work to other targets. I did not work on this one myself, but it's a very similar concept than as what we did for the GPCRs. But now we are looking at common targets for toxicity and metabolism. So, of course, we not only want to have good activity on our main target, but we want to reduce binding to some common off targets. such as cytochrome P450s. So cytochrome CYP3A4 and 2D6 are responsible for the metabolism of a large portion of oral drugs that we take. The pregnenolone receptor is a target that we really want to avoid because anything that acts on it might have similar effects to hormones, and we don't want to do that. And Herg is, of course, a target that we really have to avoid in order to reduce cardiological issues. So issues with normal heartbeat. If you hit the Herg ion channel, you might have some issues with your heart. And in order to do this, we have collected publicly available structures of these proteins. So we are lucky because in this case, we don't need to rely on AlphaFold predictions. We already have structures in the PDB. But we can still run our workflow on these targets. The only issue here is that you can see some of them, like CYP3A4 has 71 structures and PXR has 84 structures. So IFD-MD out of the box might take a little while to complete on such a large number of structures. And that's why we came up with another method, which is called consensus IFD-MD. And this is a much faster version of our tool. But instead of just docking one ligand into the binding site, we are trying to dock 2 or 3 quite active ligands, but ligands that might have different side chains or different sizes into the receptor, but with a common scaffold. And our assumption is that the core of the molecule or the chemical series should bind the same way. And the substitutions or side chains should be placed or should be accommodated by the binding modes that we predict equally well. So we can skip quite a lot of the IFD-MD workflow that I showed previously, especially the GPU parts, if we take on this assumption that the scaffolds have to be positioned the same way. So we can, we still need to run the water map and some unbiased MD simulations, which take about 6 GPU hours, but we can skip all the metadynamics scoring of the poses, which is the majority of the GPU calculation cost. And then we can just post-process the poses coming out of the W score stage with the assumption that they have to bind similarly to the protein. So this is the workflow. We have some initial structures, either from the PDB or AlphaFold. We perform FEP on the different binding modes that are generated. And if we get a good FEP model, we hope that that corresponds to the biologically relevant binding pose. If we have a non-performant FEP calculation, then it's probably a wrong pose. And then we ideate and come up with some new compounds. We put them into the FEP model and prospectively validate whether the FEP model still holds up after that. And that's with some real data in the picture. So we looked at this for the cytochrome targets. For cytochrome P450 3A4 isoform, we actually generate about 4,000 poses from IFD-MD with this very fast IFD-MD methodology. But the consensus hypothesis reduces that to only 156 poses, and only the top 10 of those we take into the FEP calculations and use the computational resources on those top poses only. And we get quite good results for CYP3A4 and 2D6. And that means that we can quite confidently predict the structures or the binding modes of ligands and how substitution of those ligands modulates the activity against these targets. But maybe more interestingly, Herg has been a target that we have traditionally struggled with trying to predict binding modes into Herg. And this is what Schrödinger is trying to tackle with this new workflow. So we have, for Herg, we have 3 protein structures. 2 of them are Cryo-EM structures, and one of them actually comes from a publication where they ran a long MD simulation on Herg and published the outcome of that simulation. So we run IFD-MD on those 3 receptors. Again, if let's say we do that with 3 ligands in a consensus IFD-MD, that means 180 poses. But the consensus hypothesis reduces that to 9 or 10 poses. And finally, we run FEP on those poses only. And there was a project that was running in Schrödinger where we had a good retrospective performance on Herg, and even prospectively our predictions held up in FEP for the new compounds. Unfortunately, I can't show structures for this project, but we actually could develop a structural hypothesis that any compound that was able to form a hydrogen bond with a certain backbone carbonyl had good Herg, like a low Herg IC50, and therefore would be probably toxic and not interesting compounds. Whereas if this hydrogen bond was not possible to be formed, then compounds were not active against the Herg channel. And with that, the project team could actually design out the Herg inhibition from the compounds. So the last slide here is that we are hoping to automate this process for other targets as well. So what we would like to do is to start from a protein sequence and the ligand set. A ligand set, and then feed this into our modeling pipeline, and our pipeline would just bring out an accurate FEP model at the end. In order to do this, of course, we need the machine learning protein structure prediction, some conformational sampling of the protein, predicting where the ligand should bind, the induced fit docking, relative binding FEP, either on a small or a full set, absolute binding FEP, and finally selecting the model to go forward with. So currently we are doing retrospective validation of this pipeline, and hopefully we can soon do some prospective validation on some interesting targets as well. So in summary, I think AlphaFold offers a great premise for structure-based drug design, but we need our best practices to be established in this space. We showed that even for virtual screening, we need some refinement of the binding pockets, but especially if we want predictive models to be developed from AlphaFold starting structures. We really need this combination of generating a protein conformational ensemble, predicting the poses with some kind of induced fit effects, and validating with free energy perturbation methods. And we showed how this works for GPCR targets and EDME targets. And we are trying to see how it works in a more generic set of targets. And of course, we are also keeping our eye on open and seeing how machine learning co-folding will pan out in the future. And with that, I'd like to thank you and happy to take questions.